Good day, ladies and gentlemen. I hope this message finds you all in good health and high spirits. My name is Raya Witaya Peron, and I am delighted to be here to be presenting my research project. Let me share you a famous quote by a Greek physician. Uh, Let food be Thai medicine, and medicine be Thai food. This famous quote, attributed to the ancient Greek physician Hippocrates, emphasizes the idea that food and herbs can have a powerful impact on health and well-being. And the quote relates to the title of my team's project, which is the development of optimized harumanis leaf extract as potential natural antioxidant agent. Without further ado, let's begin this presentation. This is the overview of this presentation, which is first, the background study, second, problem statement, three, objective, four, literature review, five, methodology, six, results and discussion, seven, conclusion and recommendation, and eight, references. So here we are at the background study. Harumanis mango, or its scientific name, which is Mangifera indica L, is a tropical fruit that is widely produced in Perlis, Malaysia. Under the Mangifera indica L group, this mango stands as a tropical treasure. Its appeal lies not only in its juicy sweetness but also in its distinctive aroma that tantalizes the senses. What makes the mango truly special are its high attractive nutritional components. Packed with flavonoids and phenolic compounds, this fruit offers more than just a delightful taste. It serves as a powerhouse of beneficial nutrients. So, there's more to the mango than its delectable flavor and nutritional value. Several studies have revealed its impressive pharmacological properties. It is an antioxidant powerhouse. The mango's antioxidants help combat free radicals, protecting our cells from damage and supporting overall health. It is anti-diabetic effects. Research has shown that components within mango may aid in regulating blood sugar levels, offering potential benefits for those with diabetes. And it has anti-inflammatory abilities. The mango's properties have also been found to reduce inflammation, providing relief for various inflammatory conditions. Following the background study, a problem statement was created. This study was devised to unravel the therapeutic potential of harumanis mango, especially from the leaves, whereby the harumanis leaves become waste when the demand of harumanis fruit increases and causes an environmental issue to the community. This research contains three objectives. The first objective being to optimize the harumanis leaves extract using maceration technique via response surface models, which is RSM. The second objective is to analyze the phytochemical of harumanis leaves extract using phytochemical and spectroscopic analysis. And the third objective is to elucidate the therapeutic potential of harumanis leaves extract using antioxidant and antimicrobial studies. From the literature review, we have found that there is lack of appropriate standards and validated analytical methods in standardization process. There's lack in adulteration and contamination. There is no standard of multi-herb formulation, and the regulatory framework of quality control standards is less to none. Therefore, uh, An alternative to improve the standardization is via optimization, whereby we enhance extraction efficiency, maximize concentration of desired compounds by minimizing unwanted compounds, and we identify key active constituents through analytical techniques. From that, we can determine the therapeutic potential of harumanis leaves extract by determining its anti-diabetic activity, its antioxidant activity, its anti-cancer activity, and its antibacterial activity. And here we are, the flow of the methodology. We start by the collection of harumanis leaf. Next is preparation of plant material and microscopic analysis. Then is followed by quality control analysis by determination of total ash, acid-insoluble ash, and total moisture content. From that, optimization of optimal condition for harumanis leaves by varying temperature, extraction time, and ratio of solvent use, which is ethanol and methanol, was devised. Harumanis leaves extract screening for total flavonoid content, total phenolic content, and total protein content was also done after that. Next was followed by chemical profiling through FTIR using prepared optimized harumanis leaves extract. Then antioxidant properties using the DPPH assay was done, and finally antibacterial properties using this diffusion method was done. So in this slide, as you can all see, this is a simple illustration of how the extraction method via maceration was carried out. Next, we carry forward to the results and discussion. As you can see, the diagram shows the microscopic analysis of 5 times magnification and 10 times magnification. This is the quality control analysis whereby the total ash content is 9.02%, acid insoluble ash content which is 1.44%, and total moisture content which is 8.93%, whereby all of the figures show that it is still in acceptable range, whereby its quality is immaculate. So in this slide, it is shown how we determine the optimal conditions when ethanol is used as a solvent. From the experimental result, we obtain an extraction yield of 24.74%, whereby from the predicted extraction yield by the ANOVA result, we obtain 22.13%. But we use the parameters that were obtained by ANOVA result, which is at 25 extraction time of 25 minutes at 45 degrees Celsius with a ratio of 42% ethanol and 58% distilled water. Due to its it is more eco-friendlier and it saves more energy because of its shorter extraction time. Same situation as the previous slide, whereby optimal condition when methanol was used as a solvent. From the experimental result, the extraction yield was obtained was 26.8%, whereby for the ANOVA result, the predicted extraction yield was 23.77%, but we use the conditions from the ANOVA result due to the fact that the ratio used was 44% methanol and 56% distilled water, which was a more eco-friendlier choice than the 50% methanol plus 50% distilled water situation by the experimental runs. So from the total extraction yield, we can see that optimized methanol harumanis leaves extract gives a higher extraction yield, whereby methanol might contain high content of metabolites. So, next is the preliminary phytochemical screening via burette test, alkaline reagent test, Elagics test, and foam test. Burette test is to test for protein, whereby yellow color change to dark purple, and alkaline test is to test for flavonoids, whereby dark green color change to light yellow green. And for Elagics test, which is to test phenols, whereby brown precipitate form at bottom of test tube, and foam test is to test for saponin, whereby foam form at upper layer of sample solution. All four tests showed to be positive results, indicated the presence of each compound in the sample. So this is the total flavonoid content results. The method used was aluminum chloride colorimetric method, whereby the sample was tested against a standard curve plotted by using quercetin standard. From the results, optimized ethanol harumanis leaf extract obtained a higher flavonoid content with 58.73 mg per gram compared to optimized methanol harumanis leaf extract. And for total phenolic content, the method used is Pauling Staccato colorimetric method. The standard curve was plotted against a gallic acid standard, and from the results, optimized ethanol harumanis leaf extract obtained higher phenolic content with 60.58 milligrams per gram compared to optimized methanol harumanis leaf extract with only 36.73 milligram per gram. For total protein content, the method used was Kedal method. 3 steps involved in this method, which is digestion, distillation, and titration. And the results obtained was optimized ethanol harumanis leaf extract or ethanolic HLE obtained lower protein content with 0.475% compared to optimized methanolic HLE with 0.6813%. Thus, for total protein content, it shows that methanolic HLE is slightly higher than ethanolic HLE. Next is the chemical profiling via FTIR analysis. As you can see here, this is the results of optimized ethanolic harumanis leaves extract. The functional groups such as the CCl bond, CCH bond, CnCO, and CH bonds are detected in the sample, thus showing the presence of phenol and flavonoid backbones justified that the extracts contain flavonoid and phenolic content. And next, this is the results for optimized methanolic harumanis leaves extract, whereby functional groups such as CH bond, CO bond, CN bond, CL bond, and C double bond O detected in the sample. This shows the aromatic phenols and flavonoid backbone found in both samples. Phenol is aromatic compound formed by hydroxyl and phenyl group. Thus, the presence of flavonoid can relate to antioxidant activities. So next, this is the result of the antioxidant activity via DPPH activity. The DPPH radical scavenging activity was tested against a well-known antioxidant, which is ascorbic acid, as a standard curve. Lower IC50 values indicated higher antioxidant activity. Thus, from the results obtained, optimized ethanolic harumanis leaf extract achieved higher antioxidant activity compared to optimized methanolic HLE. So next, these are the results of antibacterial bioassay. The samples were tested against 3 types of microorganisms, first being E. coli, Next is Pseudomonas aeruginosa, and the third is Bacillus cereus. So the positive control use were ciprofloxacin uh, and Dettol, and for the negative control were glycerin and glycerin plus distilled water. Next were the samples being tested on each of the uh, microorganisms. So from the results, the method used were this diffusion method, whereby it is determined by larger zone indicated high antibacterial properties until the bacteria cannot grow on the surface. Positive control ciprofloxacin achieved biggest inhibition zone with 3.5 centimeters. Optimized ethanolic HLE plus glycerin shown to have uh, 1.3 centimeters to 2.9 centimeters of inhibition zone. For optimized ethanolic harumanis leaf extract plus distilled water, it has shown to be 1.3 centimeters to 3.1 centimeters of inhibition zone. And for optimized methanolic harumanis leaf extract plus glycerin, it was shown to have 1.6 centimeters to 3 centimeters of inhibition zone, and for optimized methanolic HLE plus distilled water, it was shown to have 1.9 centimeters to 2.1 centimeters of inhibition zone. And as you can see here, the highlighted such as optimized ethanolic harumanis leaf extract with glycerin on Bacillus cereus has a huge inhibition zone at 2.9 centimeters, and for optimized ethanolic harumanis leaf extract with distilled water, it has shown to have a high inhibition zone against E. coli, and for optimized methanolic HLE with glycerin was shown to have a high inhibition zone against Pseudomonas aeruginosa, and for methanolic HLE with distilled water also to have a high inhibition zone against Pseudomonas aeruginosa. So, as the conclusion, the optimization of harumanis leaf extract was carried out using maceration method via surface methodology according to three parameters: extraction time, temperature, rate, and ratio of solvent used. The phytochemical compounds were determined by phytochemical and spectroscopic analysis via TPC, TFC, total protein content, and FTIR analysis. And finally, the therapeutic potential of optimized harumanis leaf extract was determined by carrying out antioxidant activity via DPPH assay and antibacterial bioassay. And so these are the recommendations that were commented for this research, whereby to use different conventional and non-conventional extraction methods such as accelerated solvent extraction and supercritical fluid extraction. And next is to further study use different parts of the harumanis tree such as the stems or the roots or fruit peels. And the third recommendation was to use high-end technologies such as HPLC, TLC, and GCMS in determination of phytochemical compounds of harumanis. Next, these are the references used throughout this project. And based on its promising insights, we have landed a partnership offer with Bisol International to further develop this research project. And that is all from me. I hope you enjoyed this presentation, and I would like to get the opportunity to wish everyone happy Ramadan Mubarak. Thank you and have a nice day.